Welcome to Precision Signals. I'm Sean Khozin. My guest today is Sush Patel, CEO of Replimune. There is a particular kind of vertigo that happens not when the science fails, but when it succeeds in ways our instruments weren't quite built to judge. Replimune engineered a virus to invade a tumor, destroy it from the inside, and expose its contents to the immune system. The idea is to convert what is immunologically invisible into something the body can finally recognize and attack. It is one of several approaches gaining momentum in a broader shift across oncology, from treating tumors as targets to be hit to reprogramming the biology around them. After granting breakthrough therapy designation, the FDA issued a complete response letter. Not a dismissal of the science, but a question about how you attribute benefit when two agents are given together in a single-arm trial. I wrote about this last summer, where I explored what the rejection revealed about the growing tension between novel biology and traditional evidentiary frameworks, and what it might mean for the next generation of combination therapies in oncology. Replimune has since resubmitted, with a PDUFA date of April 10th. But this conversation is not only about one molecule or one regulatory moment. Sush spent nearly 2 decades at Genentech. He launched Tecentriq globally, led the lung cancer franchise, and watched from inside as a company that once defined the frontier of oncology made strategic bets, some that paid off, and some it walked away from too early. He has thought deeply about what it means to build inside a large organization and what it means to leave one. He moved to Replimune during COVID and found himself navigating not just the science of oncolytic viruses, but the harder questions about evidence, regulatory expectations, and how we bring genuinely novel mechanisms forward as the science evolves, as our ability to generate and interpret evidence evolves, and as the frameworks themselves try to keep pace. The FDA's recent sounding board in the New England Journal on plausible mechanism pathways is one signal that this conversation is very much underway. What follows is a conversation about career arcs and inflection points, the evolving relationship between biology and regulation, and what it takes to lead at the edge of a field that keeps redrawing its own boundaries. Let's step into the conversation and trace the signal beneath the noise. Hi, Sush. Welcome to Precision Signals.
Hi, Sean. Appreciate it.
Great to have you here. There's a lot to talk about. You've had such a fascinating journey, and what you're working on today is quite intriguing, to say the least. But let's go back to the very beginnings, to your interest in the life sciences in general. So if I'm not mistaken, you studied microbiology at Warwick, then molecular biology at Imperial College in the 1990s. Yeah. So you were quite academic and very well versed in the nuances of the wet lab and molecular biology. Where did that interest come from? Was there something as you were growing up that sparked your interest in the life sciences?
No, sure. I was just always interested in how things work, biological processes. And if you remember at that time, it was just a very exciting time in molecular biology. We were doing the Human Genome Project. We were sort of doing a lot of genetic engineering of things like crop resistant, herbicide resistant plants, all these kinds of things. And I just kind of felt like we were on the cusp of something big. We were making advances in various molecular techniques like PCR, and it just seemed a great time to get in.
Right. It seemed like, Sush, that wasn't quite enough for you because after a few years, you made a pivot, a transition to consulting. You went to IMS Health and then Frontline. Yeah. Was there anything at the bench that wasn't satisfactory? Were you looking for something more?
Actually, I loved bench work. The thing for me was just, even when I was at college, I did a few business courses. I did some marketing. I was always kind of entrepreneurial, and at some point always wanted to sort of explore the business side of things. And I kind of realized that I wasn't going to be this scientific maven that was going to keep writing grants every 3 years. And I just felt like, I'd sort of taken the research part as far as I'd really wanted to go and wanted to explore other areas.
Right. So you were more of a big picture kind of person.
Yeah, I'd always been intrigued by the business side of things. And so I knew at some point after the first couple of years that I was going to be moving in that direction. And that's where consulting was a good transition to enable that.
Yeah. So, what was your experience like consulting? What was different from what you thought it would be, and what was the most informative lessons that you learned?
Yeah, I think for someone like me coming from science, wanting to get into business, consulting is just a really teaches you a lot of great frameworks and gives you an opportunity to just basically do a lot of different things. So what I loved about consulting is, you'd get a project, and I did healthcare consulting. So you'd be learning very quickly about a new disease area, a new asset, a new molecule, a new methodology if you were working on a launch strategy. So it was just this plethora of different types of things you were learning, whether it's disease, biology, business, analytical methods. I just love that. So, but what was sort of challenging about it is you do this project, you kind of hand it over to the client, and then you don't really know if it was implemented. Sometimes you read about it, but that was the piece that I kind of missed about consulting. Is what was
Right.
great is you do something new, you moved on to the next thing, but you never really got to see something all the way through. So that's kind of why I wanted to move on to the next kind of phase in terms of working in a company.
Yeah, no, it makes sense. You get a panoramic view
Yeah,
of the ecosystem, which is quite unique. It's very hard to get that kind of perspective from the inside of an individual company.
yeah,
I can somewhat relate to that. When I was at the FDA, I always used to say that the FDA made me a lot smarter than I am. Not because I was smarter than my peers, but because you do have this panoramic view of the drug development ecosystem.
yeah,
There's a lot of information coming to you, and you can really learn a lot, and you can, if you're curious, turn a lot of stones.
yeah.
So it seems like your consulting days served you quite well. It was a prelude to Genentech, right?
Yeah. And I tell people consulting, particularly for scientists, it's if you don't want to go and do an MBA, because some people don't want to go straight out and do an MBA. I think consulting is more hands-on, practical experience working in a number of these areas. So I think it's a great transition for scientists. And consulting also, I think, benefits from that scientific process and thinking that you've kind of established along the way. So I think there's a lot of benefits both to the consulting company, but then also for a scientist wanting to move into a broader sort of business side of things. I think it just makes a lot of sense.
Yeah, that I'm glad you mentioned that. I think there are a lot of scientists and folks in on the academic side of the life sciences that want to make the transition to something else and maybe even biopharma.
Mhm.
And it seems like consulting is a great bridge, both as a learning experience, but also I'm assuming you can build a very large network of folks that um, you can engage with, and I think this industry, despite the fact that it's so scientifically grounded, at the end of the day, it's all about people.
Yeah, exactly. It is great networking, and a lot of consultants, as you can imagine, do end up working for the companies that they consult with, particularly if
Right,
you're
right.
working in very specialized areas. There may be a kind of limited skill set out there. And so I think they're usually quite in demand for some of these organizations.
Yeah. Although I have to say, it's not for everyone.
Mhm.
If you come from like a molecular biology or scientific
Right.
background, it requires a different uh, type of thinking. One would have to shift their perspective. Is that a valid statement, assumption?
Yeah, I mean, I think it also depends on the kinds of consulting projects you get. So I was lucky. I worked in healthcare consulting, which made a lot of sense to me. I was working in oncology a lot. So even though I hadn't worked in oncology, I'd worked in more the molecular biology, microbiology side. I'd kind of had that sort of background in science. And so some of the early projects I had were very science focused. And so I think the transition wasn't so bad. I think when you get into more some of the business side, if you're doing
Right.
building models and things, that's where you need a good mentor within the consulting group. But again, how you go about the thinking around building some of those things, I was able to leverage a lot of the work and sort of scientific sort of process. So I felt there was a lot of synergies, but I think you need the right consulting framework and definitely good mentors and leaders to work with you and have a little patience as you're going through that transition.
Yep. Yep. Makes perfect sense. So then Genentech was in 2002, right?
Yep.
So you went to Genentech after consulting. Would love to learn about how you made that transition, how that came about. And you were there for almost 2 decades, which is a lifetime in this industry.
Genentech was funny. As I was doing consulting, I was doing a lot of oncology consulting, and all roads seemed to lead to Genentech in those days. I was looking at different therapy areas, and it was funny. All the assets were ended up being Genentech assets. So I had an opportunity. I had a friend who was working with me in consulting. He went to join Genentech, and then sort of said, well, why don't you come over here to do something called publication planning, which I had no idea what publication planning was. But I was so keen to just go and join Genentech at this sort of early stage that I just sort of jumped on that. And again, it was an area when I did learn about publication planning, it was a lot about science and data and figuring out how do you communicate the important information we have and generate it in a timely manner. So it was actually a good fit for me.
Right. Interesting. So is that how you started?
Yeah.
Is that considered medcoms?
It's sort of medical communications, exactly, like abstract preparation, making sure, we had many, many great scientists, great data at Genentech, but sometimes we were kind of slow in getting data out there, particularly as we were getting closer to launches for some of these publications that would take way too long. And so one part of the job was figuring out, like, how do we bring in some internal medical writing expertise? We were leveraging consultants quite a lot, but we brought in some internal people to help get a lot of publications out. And then also just how do you communicate various data at various medical meetings, which many of us work with consulting.
Oh, interesting. So you internalized a lot of the
We did,
capability.
yeah. As we were getting larger as an organization, and then there was also this move towards good publication policy, making sure, you've obviously been part of many publications, and making sure there was various disclosures, that it was really significant contribution. You weren't just adding authors for the sake of someone being recognized, that they actually had to have a significant contribution to that particular investigational trial. So those are the things that we were starting to put in place for Genentech as I was sort of
Right.
joining, and we were becoming a larger organization.
Yeah, I think people sometimes forget how important communication is as part of the drug development continuum. If I'm not mistaken, there's a company, Klein Davis and Mann, which became a global giant just because they were working on Lipitor. And that one drug really
Yeah.
put them on the map globally. So it really drives a lot of external decisions that then feed back into
Mhm.
perhaps our R&D strategy. And it seems like when you were at Genentech, towards the end of your tenure, I'm not sure, maybe you can tell us when this happened, you start to focus more on the commercial end of things?
Yeah, I actually joined. So from the publication planning role, we actually licensed in a product called Tarceva from OSI Pharmaceuticals. It was actually the first oral oncolytic for Genentech, and the sort of head of the marketing said, "Hey, do you want to come and do marketing?" And I hadn't done marketing, but they were looking for someone. We'd got this phase 3 data. It wasn't a Genentech asset. Usually, Genentech was a monoclonal company. This was something we didn't license in pretty late. It was a positive study, and they needed to get a team together. And so I joined that on the commercial side and did marketing for a few years, medical marketing, more on the promotional marketing side, and then eventually did some sales as well.
Okay, interesting. You joined after Genentech had shut down their operations in San Francisco, right? They had a very interesting lab that was working on ADCs actually at the time.
Yeah, I joined. They were still doing a lot of that research actually, and
I see.
actually had a huge pipeline. But so yeah, at that time ADCs were still very much in vogue. And if you remember, actually Genentech was a real pioneer in that space. But they were having some
Absolutely. Yeah.
challenges with the linker. Kind of keeping that linker together, and then the payload would actually, fall apart. And actually, I think they end up killing some monkeys. And it was sort of an interesting area where, even though they had this huge lead early on, they didn't stick with it. And other companies like Seagen really sort of leaned into ADCs and continued that work. And Genentech moved on to other areas.
Yeah, I learned something relatively recently that the research hub that Genentech had in San Francisco, where they were doing really innovative research on ADCs and a few other things, when they shut it down, the group that was working on the ADC portfolio went on and started Seagen.
That wouldn't surprise me at all. I think there was
Yeah,
definitely some people who really believed in it, but it's like those things in science. Sometimes it takes many, many years to have
that's
that
true.
breakthrough. And now look at what ADCs have done, right? So I remember when I first got there, we had this unbelievable portfolio and pipeline. I think that's one of the challenges Genentech had. They had such a wealth of opportunities to pursue that at the time, I think they went in another direction, and the ADC program was sort of deprioritized to some extent.
Yeah, as you mentioned, the linkers were a big challenge, and we've come a long way.
Yeah,
And I think, in a way, the life sciences is becoming an engineering challenge more and more as we delve more into the biology of oncogenesis and
yeah,
how to address malignancies at the fundamental mechanistic level.
yeah. And you talk about building. I mean, KRAS is another example of it was originally a target that wasn't druggable for the longest time, and now look at the sort of advances. And obviously, you've got companies like Revolution Medicine doing exciting things with sort of drugging those targets and coming up with different ways to tackle KRAS.
Yeah. Well, like the band, The Police, you decided to leave Genentech at the
Yeah,
height of essentially the pinnacle of what a lot of people would consider is commercial success.
yeah,
So you were the lead on Tecentriq, which was a very successful program across multiple indications. One of the first PDL1 inhibitors that really set the tone for how we view immune checkpoint inhibitors in this space. Can you tell us more about your thinking process during that time,
yeah.
being the lead on Tecentriq, and then deciding
Yeah, and I
to
should
join?
just be a little clear. On Tecentriq, so I was a launch lead globally, which, you have a global launch lead who really goes to all the affiliates, making sure they're ready. What we have in Genentech, we have this role called the lifecycle leader. And I was a lifecycle leader in one of the indications, lung cancer. So it was sort of CEO of the lung cancer program, which at the time was a very big program. And so that was kind of, there was other obviously life cycle leaders, but yes, overall it was a very big program, successful. But that was one that was interesting because Genentech was coming third to market there. It was
Hmm.
the first PDL one, but it was third to market in terms of we had nivolumab on the market and then Keytruda as well. So
Right.
yeah, it was an interesting because Genentech was usually used to being first and best, and so this was a different challenge for the company of figuring out where do you develop in a space where you're a bit late to the game. But anyway, yes, you asked the question around what made me leave Genentech, and it was a very hard decision because I'd been there a long time and done a lot of different things, and it was a very rich environment where people were very focused on development. I had a lot of great managers. You could do a lot of different things, whether it's US marketing, global, portfolio strategy, so there was, I found it very, very interesting. It was very hard to leave, but I'd reached that point where there was only so many other different roles in the company that I would really wanted to do. And I'd always, as I mentioned, felt pretty entrepreneurial, wanted to go to a smaller company where I could maybe have more decision making authority, kind of set the vision and the strategy. And at that time, we were during COVID and probably had too much time on my hands to start thinking about these things. And I kind of felt like it's sort of now or never. Once you've been somewhere 20 years, you kind of feel like if I don't make that jump now, I probably won't. And so that's what kind of led me to Replimune and sort of the biotech space.
Hmm. And yeah, can you tell us a little bit about Replimune, its origins? And it seems like obviously it's in the space of immunotherapies,
Mhm.
which is something that you gained a lot of experience in at Genentech. But what was it about the company that excited you? Obviously, the science is quite exciting,
Yeah.
but a lot of times the most exciting science is the most challenging commercially.
Yeah.
Can you tell us about the company's origins and what gave you the confidence that this is the right company for me to jump into?
Yeah, that's a great question. Yeah, I have to say, what got me to the company originally was the science. At Genentech, when I was one of the franchise heads, I was also taking on the skin cancer franchise. And one of the big challenges that we were facing pretty much across all of the indications was what to do in checkpoint failed space. And even though Genentech had a lot of assets and they were throwing everything at this checkpoint failed space, combining multiple IO treatments, but really nothing was working. And we also had BRAF, a MEK inhibition. So we were looking at different data and we really couldn't crack that nut. We were seeing, maybe we'd see that 15 to 20% response rate in that best data sets, but it wasn't particularly durable. And I saw the early RP1 data. It was a SITC presentation, only 16 patients. They had a 37% response rate. It was very durable. And what also really impressed me was the toxicity profile, because you don't want to see that kind of benefit at the expense of sort of toxicity. And that's one of the challenges when you have things like combining with cobimetinib or MEK inhibition. There's quite a lot of toxicity to deal with. So I was very intrigued by the science, so that was one thing. Although I have to say the intratumoral piece was one that I wasn't very excited about. But as I sort of explored it more, I've always been someone that likes a challenge, whether, even at Genentech, launching the first oral Oncolytic, which wasn't a Genentech product, or doing something just a bit different and challenging, maybe joining products that weren't necessarily on the, for example, Tarceva was a second to market. Iressa was the lead EGFR at the time. And so I kind of was just intrigued. I thought there was something there. It made a lot of sense to me, this idea of actually injecting a tumor locally without having that systemic toxicity just made a lot of intuitive sense. So as I started to investigate, well, why can't this be done? And so that's where my kind of commercial mind kind of thought, well, I actually think we can do this, which we can get to later on. But the other aspects of Replimune was the fact that it had a really strong management team. They had a lot of scientific expertise, and had actually sold, the first oncolytic virus and developed the first oncolytic virus, TVEC, at a company called BioVex. They sold that to Amgen. So they had this lot of expertise around oncolytic viral in general, also manufacture of oncolytic viruses. And so when Replimune was founded 10 years ago now, they took a lot of that expertise and they sort of started from scratch and actually have a beautiful manufacturing facility. It was a huge investment, but I think now, looking back on it, was the right thing to do. They also wanted to then sort of build on TVEC, because I think TVEC was a good first attempt, but I think one of the challenges with TVEC was, and you'll probably know this, is, look, 10 years ago, things like -80, the logistics, the fear of herpes, getting herpes infections, there was a lot of hurdles that the product never really overcame in the marketplace, because it was launching as a monotherapy, and it was also launching against checkpoint inhibition, which, when you've got an IV or a drug that's great versus having to go through all the logistics as unlike TVEC, it just never really took off. Then also, the TVEC data clearly demonstrated that Imlygic was a product that worked better in smaller burden tumors, earlier stage. And so I think it found a niche, but it never really found its true calling, I think. And so when Replimune was formed, the idea was, okay, these are mostly local benefit. Yes, certainly there's examples of TVEC having systemic benefit, but generally that was more the exception rather than the rule. And so they wanted to design more potent oncolytic viruses. And so that was kind of the whole genesis of Replimune. How can we do that? What would it take to sort of build on the TVEC experience? Because there's a lot of positives around the idea of local injection, the fact that you have a very good safety profile, you can get very durable benefit when patients did respond. But how can we make more patients respond? How can we also make tumors beyond the injected tumor respond to really demonstrate that systemic benefit? And so that was what Replimune was kind of formed to really address.
Interesting. And can you tell us about oncolytic viruses in general, and how you see that differentiated from this larger ecosystem of immunotherapies? Obviously, immune checkpoint inhibitors have the largest market share, if you will. We have adoptive cell therapies and CAR T cells. Obviously, you need very strong antigens and then targets to really be able to take advantage of those platforms. So how does an oncolytic viral platform fit into this ecosystem?
Yeah, I mean, I think sometimes you have to tackle, this whole checkpoint failed space, for example, is a very tough nut to crack. And I think sometimes you've got to approach things from a slightly different perspective. And what I like about the oncolytic approach is it's really a dual mechanism. And I never realized this coming in, is I'd always known viruses will induce sort of immune reaction and activation in the body, but I never realized how good they are at lytically killing cancer cells. And so, I think the challenge is, okay, it's great that you can kill cancer cells with oncolytic viruses such as an HSV1, but then how do you prevent it also sort of infecting healthy tissues? So with some modifications, they've actually been able to make it very selective for the tumor and not so much for healthy tissue. And so when you're able to do that, you actually realize it's quite a powerful local killing and lytic modality, and then that obviously releases the neoantigens that starts to drive the immune system. And then what we've really done is sort of supercharge that with then encoding various transgenes within those oncolytic viruses. And I think the big difference, particularly between TVEC, is what we've done at Replimune is one of those transgenes is GALV, which is a gibbon ache leukemia virus. It's a fusogenic protein that really creates a very immunogenic cell death. What it does is it actually clumps the cells together so that the viral replication is enhanced. And then when you've got these larger sort of syncytia, what happens is when you do lyse them open, you have even more exposure of the tumor neoantigens as well as the viral antigens, and that really just supercharges that kind of immune activation and systemic benefit that we see.
Hmm.
So that was really the idea, and it's really this dual mechanism, both the local killing as well as the additional expression of these transgenes to drive kind of the more distant uninjected responses that we see.
Very interesting. So the fusogenic component
Yeah.
essentially gives you a much more potent local effect. But at the same time, by exposing the antigen,
Yeah.
you get the added benefit of the abscopal effect.
Yeah, the fusing also makes the viral lysis and replication better and enhanced. And then when you've got more there, then you drive, even more sort of disruption of the tumor. There's more virus, there's more tumor antigen exposed. And so those things in combination then drive that systemic activation. So that's just one of the transgenes. And then our second one, that's RP1. We have got two assets right now currently under development, RP1 and RP2. RP1 and RP2 both have they're an HSV-based platform. They have GM-CSF and the GALV, but then
Hmm.
RP2 also has CTLA-4 encoded. So the idea was to drive even further up that sort of potency scale. We all know that ipilimumab is a very effective uh, immuno-oncology drug. The challenge with it's always been the sort of systemic toxicity. And so
Right.
it just made a lot of sense to actually inject it locally. You don't need a lot of CTLA-4 to do its work. But then if you actually use it locally, you can have the benefits ideally without that systemic toxicity. So that was why I think it made a lot of sense to encode CTLA-4. We also know it's a very powerful uh, transgene and an immune activator in itself.
So, kind of extrapolating from that, what are some other strategies that can be deployed this way? Beyond CTLA-4.
Yeah, we have a third asset, RP3, which when we had to make some prioritization in the portfolio, we've got on the shelf, but that encodes CD40, 41BBL. So really, there's any number of different
Hmm.
transgenes. In fact, some other oncolytic virus companies have added, PD1, PDL1 into the construct. However, I think it's interesting. I think, like many things, you reach this kind of balance of where I think the power of the platform comes, and where I think RP1 is very potent, is you want to make sure whatever you do in terms of adding extra transgenes doesn't come at the cost of the basic replication of the virus. Because if you start putting too much in there, you start seeing that the potency starts to diminish. It sounds great, right? You add as many things as possible. But I also think it's probably the right balance of the different immunotherapies or transgenes you're encoding is probably more important than just the total number.
Right. Makes sense. What are your thoughts about folks in the bispecific world that are now thinking about PD-1 and VEGF?
Yeah, we had a lot of bispecific programs at Roche, and it never quite took off. Although now I think we're seeing some of those look more interesting, actually, with Summit and other companies. BioNTech, I know, has one. So I'm going to wait and see on some of those. I think the VEGF combination, I'm still not 100% bought into, but we will see. There's clearly a lot of investment going on. And so we will determine whether that ends up
Yeah,
panning out.
I think we may find out in the near future. And yeah, once the data starts to emerge, certainly, as you said, there's a tremendous amount of interest. But I think the common thread between those efforts and what you're working on is to really think about a pathway directed approach versus just targeting a single antigen and having a more rational way of addressing the oncogenic process. So thinking about that rational approach, obviously it requires a lot of data to uncover the fundamental mechanistic realities
Yeah.
of tumors that are quite heterogeneous. There's macro environmental factors that are involved. How do you navigate this complexity in terms of focusing on what matters the most, mechanistically speaking?
Yeah, I think what we're doing now, and actually we're almost sometimes going backwards a little bit to go forwards, because I think sometimes, with small companies, you rush to the clinic and we've I think generated some great data, but we really want to understand what's going on biologically. And obviously the bigger company may have done that in a more stepwise process. So things like, going back and looking at gene signatures, seeing things like clonality, what new clones are we actually expressing? Sometimes we see an expansion of existing T cell clones. What's interesting is we're also seeing the expansion of new T cell clones and new gene signatures. And so we're almost going back and going back to understand that. We are working with the Institute of Cancer Research in the UK. There's some historical with Kevin Harrington and others. The company, even back from the BioVex days, had a relationship with Kevin, and he's got a really good lab. So they're definitely going back and sort of understanding some of the underlying biology. One of the things that I think was quite interesting at this year's SITC, we had a late breaker looking at really the tumor microenvironment, day 0 and day 43, to see what's changing, because you always get this question, which we can get to later on, is what is the RP1 contribution? And obviously we believe we see that clinically, but biologically, can you also help to explain that? And so what we see at day 0 is really the prior checkpoint doesn't really do anything to the tumor microenvironment. It remains pretty cold. Day 43, when the RP1's on board and had a chance to do its stuff in the tumor microenvironment, we really do see that it becoming more inflamed and various gene signatures starting to really manifest, particularly in responders. So the idea that you're really seeing this differential impact of RP1, and then I think that's allowing the checkpoint then to do its work again.
Right.
So that's why I think and going back and sort of understanding that is really important for us, and we're doing some work on that.
Yeah, that's very interesting. So that would open a new array of indications, sort of the cold tumors that have been historically so hard to tackle. Definitely looking forward to delving into that. And obviously, you are essentially the poster child of precision oncology. You are very much developing bespoke therapies for patients that have very clear intrinsic mechanism behind their malignancy. There's been attention, I think, in cancer drug development between having a bespoke precision oncology approach versus an all-comer approach. How do you balance the two? And where do we think we're going as a community, as an industry?
This is really interesting, because this goes back to my Genentech days as I was leading the rare cancer franchise. But I remember, and you'll remember this, back in the day, I remember EGFR, a lot of drug development was all about all comers. And, people were very worried about going to only 10 or 15% of the population with EGFR in sort of Caucasian patients, because, oh my gosh, there'll be no market. Now you look at drugs like osimertinib, it's a $7 billion drug. So I always found this amusing, that I think finding the right drug for the right patient and having great outcomes means they're going to stay on drug longer, they're going to do better. There's a commercial case there, clearly. And so, ALK was another great example, 3 to 7%. And one of the products that I had the great fortune to work on was Alecensa, a great ALK inhibitor. That's almost a $2 billion drug now, right? So it's funny that you had these small prevalence, but lung cancer is a big disease, and so I think you can be very successful with that. The idea of, precision, to me, I still believe that finding the right drug for the right patient is important. It's interesting. We haven't been able to find those biomarkers with oncolytic viruses so far. We've looked, and there isn't a clear biomarker signal for us to predict who's going to benefit. I think some of that is because of the dual mechanism. You've got that local killing. I think it's quite hard to sort of predict ahead of time from an oncolytic viral perspective of who's going to benefit.
Right. Obviously, we talked about tumors that are not immunogenic in general. So it seems like there is a way to address and tackle the tumors with oncolytic viruses. But it is interesting that no biomarker has surfaced. So do you think that ultimately it's about orchestrating an immune response, and the specificity comes from how you orchestrate that?
I think that's probably right. I think we want to get the immune system going, and then maybe Some of that could be the oncolytic virus sustaining that, but then it could also be the combination partner then being allowed to do its thing. And so I think that is right. I think that's what we fundamentally do. I look at it as very much really enabling other drugs such as checkpoints to work again. Well, that being said, what's interesting though with RP2, we do see in uveal, which is an immunogenic, it's a
Yep.
very immune insensitive tumor. It's a small data set, but we have seen some monotherapy activity with RP1, which we haven't really seen with RP2, which we haven't really seen with RP1. Now, is that the CTLA4? I suspect that could be having a role. And so the question for us as we do more analysis and work in uveal is there a role for monotherapy as well as combination? The trial we're doing is in combination, but certainly I'd like to generate some monotherapy data with RP2 because that may actually, there may be a group of patients that you don't need to actually use the checkpoint in.
Yeah. What are the solid tumors that are on your list in terms of being most amenable to oncolytic
Yeah,
therapy?
the way we've gone about it, Sean, is I think we've sort of initially built on, we know skin is in a very sort of genic sensitive tumor. So we've done a lot of active work in, melanoma, non-melanoma skin cancers. We're actually looking at monotherapy with RP1 in solid organ transplant patients that develop non-melanoma skin cancers. Many of those can't get checkpoint inhibition because of the risk of organ rejection, although, people do use them in liver transplant patients because you have dialysis to fall back on. But what they don't tend to do is heart, lung, because at that point, getting another organ is very, very challenging, whereas we've actually been able to treat those patients and haven't seen any organ rejection. So I think that's exciting. So there's a lot of organ transplants done in the US, and for those non-liver patients, there really isn't great options. So we're pretty excited about our monotherapy data in Articus, but that's coming. As we think about other indications, the other aspect that we didn't really get into is, I think we talked a little bit about the uninjected responses we're seeing in visceral organs, liver and lung. And so I think this naturally an interesting area, and we know a big challenge in oncology is when patients develop lung and liver metastases in general. And so the challenge, though, has always been with an intratumoral. Well, these superficial skin lesions are pretty straightforward to inject. How are you going to inject these deeper lesions? And that's where, when we talk, going back to kind of our conversations, I came in commercially, we've done a lot of work with interventional radiology. And this is something they can absolutely do. I think technology's got better. We've got far more sophisticated procedures that they do, like embolisms, TACE, tear. So they can absolutely do repeat liver and even lung injections using both
Right.
ultrasound and CT in a pretty routine way now. And we've generated more than 1,000 actually 1,200 deep injections across our program. And we have more than 800 in liver and lung cancer now, seeing a very acceptable safety profile, seeing that we can inject even the lung, 4, 5, 6, 7 times. Even though we do get some pneumothorax, it's self-resolving. It doesn't prevent these patients being able to continue the treatment all the way through. And so, but what's amazing is we're actually seeing really good responses in those patients, because I do think liver is kind of an immune sink, and in itself is an immunosuppressive environment where a lot of checkpoint drugs don't work very well. So our direct activity in the liver may actually enable that to become susceptible to the checkpoint inhibition. So that's why we're quite excited about taking liver and lung metastases and ultimately primary liver and primary lung lesions as well, and injecting those. So kind of our development program is building on what we've seen in skin with these liver and lung metastases to uveal, which is almost 90%, by the way. 90% of uveal, roughly, patients will develop liver metastases. And in our trial, nearly all the patients are injected in the liver. So we're building that expertise, working with IR, and then actually now translating that into, we have an HCC cohort in atezo-bev failed patients, and we've just opened a biliary tract cohort as well. And this is more primary liver and biliary tract injections.
Do you envision a scenario where you would pursue an indication for any metastasis to the liver? Obviously, metastatic disease in the liver can be quite catastrophic.
Yeah.
One of the primary reasons.
You mean like a basket, almost like a liver basket trial or something like that?
Yes.
Yeah, we've thought about that. Part of the challenge a little bit is I think it makes a lot of sense, and this is an example where I think we'd have to have some regulatory feedback, is in different tumors, Obviously, the standard of care is different. The outcomes might be different. And so I think it makes a lot of sort of rational sense, but how do we actually operate and execute that? And how do we then determine whether the efficacy we're seeing would justify a liver agnostic indication is
Right.
something that I would need a little bit more thought. But yes, in general, if you look at the prognosis of liver and lung patients across all tumors, they tend to do a lot worse.
Yeah, in terms of having a high unmet medical need is
Yeah.
certainly a major challenge. And as you alluded to, I'm sure there are lessons learned from the tissue agnostic approvals that we've seen in recent years.
Yeah, we've certainly, I mean, Genentech again, we were fortunate enough to have look at things like ROS and RET, which quite rare prevalence in some tumors like lung, but when you add them up across different tumor types, I think it's pretty interesting. We're seeing that with HER2 as well in some diseases as well. So I'd like to see that continue again. Even some of the immunotherapy agnostic, in MSI, was it MSI high? I think it was the MSI high. Was also interesting as well.
Yeah, yeah, and this is where it's mostly about developing, I guess, a compelling evidence package to convince folks, because there's no mathematical threshold that one can define. Um, for example, most, if not all, tissue agnostic approvals have been given to therapies where they didn't test every single tumor type. And in cases that there were rare tumor types in the mix, then may have been one or two cases.
Right.
Well, is that enough? Who knows? It's in the eye of the beholder.
We don't know. I mean, interestingly, one of the areas we're thinking about is sarcoma subtypes, checkpoint failed sarcoma subtypes, which that's a lot of different tumor types when you look at sarcoma. But I think when they fail checkpoint, they all tend to do pretty poorly. And so once they've had chemo or they've failed checkpoint, there isn't great options. So is that an opportunity for us? So that's one of the areas where we've definitely seen some activity in sarcoma with RP2, and actually interestingly RP3, which we previously had put on the shelf, but had seen some actually pretty interesting activity with RP3 as well in sarcomas.
Interesting. Sush, you've made a very interesting decision across your portfolio not to pursue any big pharma partnerships. Can you walk us through that calculus?
Yeah, I mean, we have actually worked closely with some pharma. So BMS, for example, has provided drug in the Ignite trial for nivolumab. Roche is providing atezo-bev. However, and then actually we were working, Regeneron in CSCC and ASAPOS trial was actually not only providing drug, they're actually co-funding that trial. So we have had collaborations, but we've actually not wanted to give away too much too early. I think the company's been very fortunate that they had a good funding situation. And because, as I mentioned, the founders had a lot of experience with, TVEC and working with other partners, they didn't want to give away too much too soon, because I do think we've always felt in skin cancer, at least, this is something we can very much do ourselves. It's a pretty small community. I think it's something that we could take on ourselves. And also with the intratumoral piece, Sean, we wanted to make sure we do this right. We're kind of a prove it story, because the reality is intratumoral agents, particularly as you think about these deep injections, have not been commercialized successfully.
Right.
We believe we can do it. We've done a lot of research, and if we get the opportunity with approval, we'd like to sort of demonstrate that. We don't want to just hand that off to a pharma partner, because I think, as we know, pharma does certain things very, very well, but generally they do what they're comfortable with and have already sort of established. And so we want to try and get that going before bringing a pharma partner on, particularly in skin cancer. Now, we don't have the bandwidth and expertise as we think about going to large indications, HCC or GI indications. That's where I think we would want to bring a development pharma partner on. And at that point, hopefully we've got more experience commercializing these things that we could then translate that into successful commercial launches with a partner globally.
I see. Because as an outsider, you're kind of looking at how you've been conducting these partnerships. It seems like, you have retained full control of the commercial rights. Is that something that you foresee to be one of your main criteria?
Yeah, I think, like I said, for skin cancer in the US, yes. Now we have made a decision. We're not going to go outside the US. So if we do filings outside the US, then yes, we will very much seek a commercial partner. But in the US and in skin cancer at this stage, we're planning to go it alone. That being said, of course, if the opportunity comes along for the right partner, that's certainly something we could discuss.
Let's move on to the FDA, Sush.
Yeah.
The regulatory journey of your company has been quite fascinating. You had breakthrough therapy designation. You had alignment through mid and late cycle reviews at the FDA. And then all of a sudden you received a complete response letter from the agency. Can you tell us more about that and how that came about, and how did you address it, and what's the strategy as we speak?
Yeah. Well, I mean, it came, literally the day before our PDUFA. So yes, it certainly caught us off guard. We were expecting an approval and a final label. And so I think getting the CRL was a shock to the system, particularly as we hadn't really had any indication before that. We had late cycle, mid-cycle meetings where, AdCom wasn't necessary. None of these topics were brought up. But as you know, the FDA had gone through various changes and division changes between OCE, CBER, and then also leadership changes. And so we don't know exactly what happened, but clearly there was a change in perspective on the data, whether it was new people coming in, looking at the data. So, I think what we've done since is really the reasons for the CRL cited were heterogeneity, contribution of components, and there was some concerns with our phase 3 confirmatory trial design. So we had the Type A meeting, and I think a big part of that Type A meeting, it's also kind of going back and re-educating people on why we did what we did. I mean, I think we understand, and I think you'd appreciate it. We all want to do a randomized trial, but there are settings where it's actually very, very challenging. So I think explaining to the agency why we did what we did. We also had some of the top KOLs in melanoma come to the meeting and work with us to explain why in a definitively failed checkpoint, patient in melanoma, further monotherapy PD-1 would not really do anything. And so we had them explain why. Then forcing people to do that in a phase 3 is just not ethical. Yes, it's one of the options in the dealer's choice because it's an NCCN listed option, but in a very, very limited situation. And actually, we explained to the agency in the trial to date, because we know what physicians choose in the dealer's choice for the randomization, no one, surprisingly, has actually received monotherapy PD-1. Nearly all the patients had actually received Opdualag. So I think the agency got comfortable with the phase 3 design, with all the KO and education around why we did what we did in terms of a single-arm trial, a definitive criteria. I do believe that helped. As we did the resubmission, we also submitted some additional information to the agency to further address their questions. My view is the agency has believed the drug was active, and I think when you've got a combination product in a single arm, it's what's that level of contribution. And so while we think the criteria we use should get you there, because if you use that strict criteria, really further monotherapy really doesn't do more than 5 or 6%. Even combinations like Ipi-Nivo or Opdualag get you around 10%. But even those data and trials didn't use the same strict criteria. So that should give you some sense of the contribution. But what we did to further help the agency is we used patients their own control. And so what we did is actually looked at the response rate for patients before they came on to the IGNITE trial, whether that was combination treatments, single agent PD-1. And what we showed is if you look at outside the adjuvant patients, because we had about a third of the patients were adjuvant, you obviously can't look at their prior response because they didn't have any tumor. But if you look at the non-adjuvant patients, they had about 11% response rate coming into the IGNITE trial. Now, just as a framework, and you would know this, frontline melanoma patients, typically the response rate is 30, 40, 50%. So when you have 11%, firstly, that would tell you it was already a group that wasn't doing very well. They didn't respond very well to their prior treatment. Then when we looked at those same patients, we got a third of them into response, in durable response. So that was one of the pieces of data we sent. We also showed that for the patients who were primary resistant, who failed their treatment in less than 6 months before coming on, they had a 0% response rate to their prior treatment. And we also got about a third of those into response. So when you look at even the difficult subgroups, using patients their own control, I think it further kind of highlights the contribution of RP1 in these definitively failed patients coming into the IGNITE trial. So Obviously that was one of the pieces of data and some other information we submitted. So right now they're reviewing the information, and that review is ongoing, and we hope that it's sufficient to get them over the line and address their concerns.
Very interesting, Sush. So that was part of your resubmission of the BLA, and there's a new PDUFA date of April 10th,
Correct.
right?
Correct.
Yeah. This is a very rational approach to address some of the questions that the FDA had raised. In fact, it's very much compatible with the commissioner's thinking and also the head of CBER's thinking. I'm thinking about the sounding board that they published in the New England Journal of Medicine back in October on a new pathway, which is called the plausible mechanism pathway.
Exactly.
It seems like the FDA is open to considering mechanistic rationale in lieu of doing, say, a randomized study, because the patients themselves can serve as their own control. So do you think that pathway opens up new opportunities for addressing complexities in running big studies where the populations are small? And if so, how would that change the accelerated approval pathway? What would be the ideal scenario in your view?
That's a great question. I mean, we're hearing a lot around, I think, flexibility around rare disease, small populations. And as you and I know, cancer is a series of very rare subpopulations. And so it would be wonderful if we could think about using this approach in these particular difficult settings where we haven't had approvals really, or very few approvals. Checkpoint failed disease is a great example. I mean, we've been on the market with checkpoint inhibition more than 10 years now, and I'm not very aware of many actual approvals we've had beyond TIL therapy in melanoma, specifically in checkpoint failed disease. I'm sure there are a few, but not many. And so I think we desperately need more options for these patients because it's one of the biggest unmet needs. And the idea of using this plausible, sort of rationale makes sense because I mentioned even the biological plausibility. As I mentioned, if you've got biopsies and you're changing the tumor microenvironment, that should also suggest that clearly this is not happening by magic. RP1 is clearly doing something to allow the checkpoint to work. So I would hope it's a combination of biology and then using things like patients their own controls that we could actually adopt this for oncology, and that hopefully oncology is not treated differently somehow, because I think the unmet need's just as high. Melanoma patients are dying every day, unfortunately, when they run out of options, just like some of these rare disease populations where there isn't great options or no options. So I would hope that would be a mechanism that we could discuss with the agency. And then you talked about the accelerated approval path. To me, one of the challenges we have even now in our confirmatory trial is we're doing well, we're enrolling well, but the reality is people aren't excited about enrolling patients in the control arm.
Sure.
Even today. And I think there are examples where the accelerated should just be the full approval. To me, when we've talked about it, the commission has also talked about sometimes needing one trial. Now, that's a one randomized trial, but maybe in these areas of high unmet need with plausibility and in the right setting, that there is openness to just using the single arm data as the approval, full approval. Because I think forcing patients to do some of these confirmatory trials is challenging. Now, I understand certainly that might result in some things moving forward that perhaps are more marginal in benefit. But in general, I think if you look at the accelerated approval pathway, I think there's been more positive developments as a result of it. I mean, Pembro was originally approved as a single-arm accelerated approval option. And so I think there's always risk with that. And I understand we want to maintain high standards, but I would hope that there's some discussion on, okay, here are the settings where we are comfortable. And maybe that's an early discussion we have with the agency where something like this could apply, whether it's sarcoma. I mean, they've also talked about diseases like sarcoma where they want to see more drug development. So maybe there are certain settings, certain areas, certain disease types where there is that openness to do things a little differently. Whereas maybe, look, I understand if it was like frontline lung cancer, yes, you're going to want a randomized confirmatory trial. But that may not be the case in all other settings. So I think that would be a great discussion for us to have.
Well, the FDA has used single-arm studies for full approvals in the past.
Of course, yeah.
There has been a compelling mechanistic rationale, or in one case I can remember, the FDA approved crizotinib in patients with an ALK1 rearrangement based on a single arm study of 50 patients. Because by that time, everyone knew about crizotinib,
Right.
were testing for the ALK mutation.
Yeah.
It was actually in the NCCN guidelines. So it was almost routine practice. So that level of confidence and evidence, when that exists, whether it's coming from the clinical experience of folks using the drug essentially off-label,
Yeah.
or through a bottom-up approach, a mechanistic rationale that's strong enough, FDA does have a legal mechanism to approve those therapies. I personally was very encouraged when I read the plausible mechanism pathway sounding board. Obviously, it's not the end of the story, but I think it's a new beginning that the community should really take advantage of. I think we also today have the ability to gather efficacy data from the real world. We have tools available to us that we didn't have even 5 years ago. So all of those realities combined tell me that the FDA is certainly on the right path. But I do think the biotech community is stepping up to the plate.
Yeah. Oh, I think we need it because I think one of the reasons Replimune came to the US, it was actually founded in the UK. The reason they came to the US was one, obviously investment and trying to make sure that they could raise money. But the other one, and the big part of it, was the regulatory environment.
Right.
It allows biotech to be creative. And I think that was always encouraged. And it was something that pharma, doesn't always do well. They kind of do things in certain ways. And I think they very much look to the biotech industry to come up with unique
Yes.
and different ways, take that risk on. It is a high risk, high reward kind of approach in business we're in. But I also think that accelerates options for patients. And so I would really hope that we don't lose that, because I do think that otherwise we risk innovation going to other countries. And I know that's a big theme right now, whether it be China or other places where I think they are willing to try different things. And I just hope that we, in our need for obviously high standards, things like randomized data, which I think again we all believe makes sense. I hope we don't lose that opportunity to think outside the box and think of creative ways to get drugs to patients faster.
Yeah, I completely agree. So you do believe that your colleagues in the biotech universe are stepping up to the plate and assuming that risk, which means you think our big pharma colleagues should be a little more bold and venturesome?
Yeah, I mean, I don't want to suggest that pharma isn't, I think certainly there's many pharma companies have done bold and interesting things, but I think sometimes they actually are looking to the biotech. That's exactly kind of how this ecosystem really works very well. I think they do things in a certain way. They do certain trial designs very well. And I think they actually encourage biotech to do that. And I think that's why, I think biotech often thinks about things very differently. And that's what I think is what's exciting. They tackle the problem from a very different perspective, from a small company perspective. And that's what I think has been really successful for the US biotech industry in many, many examples. So
Yeah,
I hope
it's interesting to hear that from you, because mathematically speaking, one would assume that for biotechs, really messing with novel clinical trial designs and evidentiary standards is actually the least palatable thing, because they're taking so much risk in the asset itself.
Yeah.
But I guess it's the culture of these companies that drives them towards more innovative trial designs and evidence packages. So it's really, again, going back to, it's all about the people and the culture.
And I hope that the regulatory environment, again, I don't know how we sort of have these discussions up front so that there's consistency, because when there's consistency, I think what we'll see is investment in biotech from a lot of companies and investors we talk to. There is a little concern on their end now, because they would make those investments. They always realize there was risk, but there's risk in just the drug itself. There's a risk in the clinical trial. I mean, there's so many other existing risks. If then the regulatory risk is very, uncertain, even after you've made certain commitments, I think that will be really difficult for the industry. And so hopefully we can get to a good medium and compromise, and understanding of what are the benchmarks, what are the rules, what do we need to demonstrate, so that we can all, work towards getting what the FDA requires.
Yeah. My observation, having been at the FDA for a few years, is that the FDA typically hesitates when it comes to defining new frameworks and new standards. And for good reasons, because they don't want to make top-down decisions that have unintended consequences. So they rather go with community consensus. And a lot of these issues that we're talking about, Such, there's really no community consensus. So it becomes a very difficult job for the regulator to suggest and recommend a new pathway or a new framework where the community itself is not harmonized in their thinking.
So it sounds like what you're saying is that some of this is on us to kind of help.
I think it is. Yeah, and if we think about all the standards that the FDA uses, for example, the RECIST criteria for tumor measurement, it was really the community consensus that drove the FDA to adopt it.
Right.
There are many people at the FDA that really don't really speak fondly of the RECIST criteria, for example. But it was something that the community rallied behind, and it really drove the FDA to adopt it as a quote unquote standard, even though they never talk about it in terms of standards. FDA, technically speaking, does not have the statutory authority to come up with top-down standards. It really has to align itself with best practices and the leading edge of science. And I think on these questions, really the moral of my story is that we may have an opportunity to really develop consensus around these themes that can help the regulators improve how they advance therapies for patients with high unmet medical need. Because the current framework, frankly, is no longer compatible with the science that we've been advancing for the past, frankly, decade.
Yeah. Well, also, I mean, outside just the feasibility of doing it, I mean, the cost now for small companies to do randomized trials is astronomical. The inflation rate we're seeing, just like everything else on trial costs, is just exorbitant, and it's just increasingly hard to enroll these studies, to pay for these studies, and so I do fear a little for that in the current environment.
This is almost a separate discussion, but I think we need to honestly and mathematically look at the existing standards of how we assume efficacy and look at effect size in a randomized study. We really are looking at average treatment effects, essentially an abstract theme that doesn't exist in the real world. There's no average patient.
Right.
And we assume that, everything around it is noise. Whereas, no, it's not really noise. It might be actually heterogeneity.
Yeah.
What statistically we call noise. So I think there might be an opportunity to revisit some of the fundamentals of how we assess efficacy.
That's interesting, because even randomized trials aren't always a gap. I mean, at Roche, we had many randomized phase 2 trials that looked very, very encouraging, that never translated into positive phase 3 studies, whether it's METMAB, TIGIT. There was also a number of examples, I think, out there in oncology where we see this. Sometimes the randomized data doesn't actually translate.
This is a statistical dogma almost that, we are so focused on the average median effect size and treat everything else as noise. And you're right, sometimes it actually leads people in the wrong direction. Going back to gefetinib, that's an interesting story, right? Because the confirmatory trial failed, even though the accelerated approval showed efficacy of gefetinib, which is an EGFR inhibitor in patients with lung cancer. Well, we didn't know that the driver was actually the EGFR mutation. And so when the confirmatory study failed, there were patients that were responding. In the
female adeno never smokers who
it turns out that they had a clinical feature. Right, exactly. But that wasn't noise. It was the driver was EGFR. So once we figured that out, well, unfortunately, it was too late for gefetinib, but erlotinib, your tech study really put EGFR inhibition on the map for patients with lung cancer. But it was a mechanism, the scientific understanding that drove that forward versus our statistical assumptions. But again, separate discussion. Looking forward to having side conversations with you on this because it seems like you have a lot to say about the statistical assumptions we make in randomized trials. So, Sush, time goes by fast when you're having fun. We're almost out of time, and there's a few other things to talk about. I'd love to get your thoughts on immunotherapies in general. Where do you think the field is going? We have the approaches that you're taking. We talked about immune checkpoint inhibitors, cell therapies. Where do you think the field is going, and where do you think we'll be in, let's say, 5 years?
Mm. Yeah, it's really interesting, and I think in some ways we've reached a plateau with immunotherapy. Whether we've reached that kind of ceiling on the upper end with frontline studies. Although I think in neoadjuvant adjuvant we're seeing amazing things, so that's great. We're curing, I think, a good group of patients, and I think we'll see more and more neoadjuvant adjuvant studies that'll report out positive. Unfortunately, we know there's that group of patients still who don't benefit and progress quickly, and they do just as badly as metastatic patients. So I think figuring out this, going back to figuring out what is a secret formula, and I think there'll be a lot of effort and drug development effort put on these failed, checkpoint failed patients, whether it's adjuvant failed, frontline failed, and addressing that. And is that combination, how are we going to tackle that? Is it combinations? I don't think the IO combinations have really panned out. I think throwing further IO at these patients doesn't seem to be it. So I don't know whether there's other combinations where what we haven't been successful so much in is if you think about targeted precision medicine combining with IO, just hasn't seemed to work out very well so far. Maybe that'll be different with RAS. We'll see whether that's the case or not. Some of those studies are going to be coming out. So I think figuring out the right combinations in these populations. And yes, I think maybe other approaches. I mean, some of these personalized vaccines, I think, haven't quite got there, but they you wonder at some point, are we going to figure out how do we drive that sort of single agent or that sort of really strong immune response? I think we haven't. I don't know if we've quite got there yet, but I think we're getting closer.
And so, what are your thoughts about the emergence of AI in drug discovery and development? Where do you think we are today? And do you think it's something that can truly make a difference in how we develop new therapies?
Yeah, I think it's very early. I do think, just like everything, there's a lot of hype right now, but I do actually think it will ultimately translate into the promise. I think it's just going to take us a little bit more time than where I think we're currently. I think we have a little unrealistic expectations, but this happens with many new technologies, and I'm actually optimistic we will get there, and I'm optimistic AI will play a big role. I'm not sure we've figured out exactly the role AI is going to play right now, but I think we will get closer to that as we continue to invest. I know some companies are putting a lot into it, and for us right now, at least at the small biotech side, we're kind of unfortunately waiting for others to lead a little bit and sort of following rather than leading the charge. Although I'm sure there's other smaller biotechs that are probably being more aggressive and taking this head on.
Sush, we're unfortunately out of time. Thank you so much for your time. This was a fantastic discussion.
Yeah, I appreciate it. It's great chatting to you, Sean.